Hi, everyone. Thank you all for joining us for our April Research Spotlight webinar. I'm Dr. Sarah Hernandez, the Director of Research Programs at the Hereditary Disease Foundation. I want to start by thanking our sponsors for this webinar, Neurocrine Biosciences and Spark Therapeutics. Also, thank you to everyone who supports the HDF. Your donations are used to fund the research that we're featuring today, so thank you. Today's webinar covers an exciting topic that I know there's a lot of interest in, and that's gene therapy. There's a lot of buzz around this topic because it could be a real game changer for developing one-time treatments for a lot of diseases, which includes Huntington's. Today, we're going to hear from the team that was awarded an HDF Transformative Research Award in 2023 for a project that focuses on advancing gene therapies for Huntington's. This was an award for $1 million given over 2 years. And the goal of this project is to use a harmless virus to deliver a therapeutic that targets modifiers of HD. This is a preclinical study that's being done in monkeys and, if successful, could lead to clinical trials in the near future. We're joined by the lead PI from that award, Dr. Bev Davidson, as well as key members of the team, Dr. Leslie Thompson and Dr. Jang-Ho. Dr. Davidson is a professor at the Children's Hospital of Philadelphia and University of Pennsylvania, where her lab focuses on genetic diseases that affect the brain, investigating how mutant gene products contribute to disease and why certain brain regions are more susceptible than others. Her team employs advanced molecular methods, sequencing and imaging in animal models, and a variety of different molecular tools to test various hypotheses. Her lab is also developing next-generation therapeutics for inherited disorders, including the engineering of novel gene therapies for tissue and cell type-specific treatments. Dr. Thompson is a professor at the University of California, Irvine, and she has studied Huntington's for almost her entire scientific career. She was a member of the HDF Venezuela project that identified the causative gene for HD in 1993. She's trying to understand how the HD mutation damages brain cells and identify targets for new drugs to prevent or improve that damage. Dr. Thompson has also worked with a group of investigators to establish the HD Patient-Derived Stem Cell Consortium and is using stem cells to study HD through a multi-institutional collaborations and big data approaches. She's currently evaluating the use of human neural stem cells as a possible therapy for HD. Dr. Cha is the Chief Scientific Officer at Latus Bio. Previously, he was the Global Head of Translational Medicine Neurosciences at Novartis. He obtained his MD and PhD from the University of Michigan and did his residency in neurology at Mass General Hospital in Boston. During his career at Mass General Hospital, Dr. Cha held multiple positions, culminating in Associate Professor of Neurology. Thank you all so much for joining us today. We're excited to hear about your work. Before we start, I want to remind our audience that you can ask questions throughout the talk in the Q&A box. So be sure you use the Q&A box, not the chat box. Feel free to ask whatever question you might have for our speakers, technical, non-technical. We'll hopefully have about 15 or 20 minutes at the end for a Q&A with our speakers. So send those questions in as you think of them so that you don't forget. Also, this will be recorded and available on our website and YouTube channel in about a week or two. So if you want to watch it again or if you want to share it with someone, it will be available for that. Thank you again, everyone, for being here, and I will pass it over to Bev.
Okay,
great.
Great. So I'm going to go ahead and get started for this morning's HDF webinar on developing transformative tools, gene therapy for Huntington's disease. So this is a little presentation from myself, but I'd be remiss to say that it's my work. It is definitely a team effort. And I've included a fairly recent photo of the lab here, including some individuals that have transitioned to Latus Bio, as you'll hear later on. So first of all, who am I? I'm one of the only of the three of us I'm the newest comer to HD research. Where did I start out? I grew up in the plains of Nebraska, and this is truly what it looks like there. Wide open spaces, and yes, we really do have these sorts of storms. But I grew up in a really fantastic, small, but amazing community of individuals. And the entire community was very excited about medicine and education and promoting healthcare. And in fact, my dad, whom they named this clinic after, was a large part of my interest in spurring my interest in medical research and the development of drugs for patients. And in fact, he was the one that introduced me to Huntington's disease when I was a child, as we have a family in this small town in Nebraska afflicted with Huntington's disease. I wanted to be a marine biologist, and so I had applications for undergraduate schools, both in California and Florida, but my father and mother said, no, you have to stay in the state of Nebraska. So I went about 180 miles to the east to Nebraska Wesleyan University, where I got my degrees in chemistry and biology. After that, I went to the University of Michigan, where I obtained my PhD. Just to give you a sense of the timing of this was when Jang-Ho and Ann Young were actually at the University of Michigan. However, I did not know that at the time. But what I did know is that this was a great time for University of Michigan basketball. After my PhD and some postgraduate work at Michigan, I was recruited to the University of Iowa Hospital and Clinics in Iowa City, Iowa. And there, my husband and I raised our young family for over the course of the next 20 years. About 10 years ago, I was recruited to the University of Pennsylvania and the Children's Hospital of Philadelphia. And from there, I've been working on advancing next generation therapies for HD and other brain disorders. So what is the Davidson's lab journey into HD and HD gene therapies? So as Sarah mentioned, I've been interested in developing novel tools for the brain. And this has been happening since the 1990s, but really it was in 2000 at the University of Iowa where I made a concerted effort to advance adeno-associated virus gene therapies for the brain. And we embarked on some work on RNA silencing after Andrew Fire and Craig Mello had won the Nobel Prize, and we're the first to vectorize that and show that it could work in animal models. And we published that work, and then soon after, I got a call from Nancy Wexler to come to an HDF meeting in Boston. And it was there that I had experienced the famous Nancy Wexler hug and was forever hooked and drawn into the field of HD research, as well as advancing HD gene therapies. And shortly after that meeting, we applied our technologies and were the first to show that this could work in mouse models. And then over the ensuing years, we worked to improve upon our vector systems for gene silencing for HD, even going so far as to testing these in large animal models, including monkeys. In 2011 and 12, I started collaborating with Leslie Thompson, and our first paper was published in 2013. And this is really one of the fundamental questions we hope to address in this Transformative Research Award. 2014, I moved to the Children's Hospital of Philadelphia in Pennsylvania and continued my work with Leslie and others, and also really initiated some of the discovery work that has led to the foundational efforts of Latus Bio today. And then, of course, in December of 2023, we received the fantastic news that we had won the HDF Transformative Research Award to advance our ideas. Why did I move to the Children's Hospital of Philadelphia and the University of Pennsylvania? And it's really quite simple. The opportunities at CHOP continue to be unprecedented in advancing gene therapy-based approaches for any number of disorders. And this was all started by Kathy High when she founded the Center for Cellular and Molecular Therapeutics, or CCMT, which is now endowed by the Raymond G. Perelman Foundation. And so it's the Raymond G. Perelman Center for Cellular and Molecular Therapeutics. And what CHOP CCMT provides us are incredible core services to advance our ideas from the preclinical to the clinical stage. And this includes an animal model services core that supports preclinical testing and pre-IND work, state-of-the-art manufacturing for vectors for gene therapies that actually go into patients. Two of the most early approved gene therapies actually arose out of this clinical manufacturing facility. We have regulatory support for interacting with the FDA and European agencies, and then a clinical support team that helps manage our movement from the bench to the bedside. So what is gene therapy, just in a nutshell? Gene therapy can really be defined as the process of adding genes to cells. And this could be done for several reasons. One is to replace a missing gene. One is to inhibit the expression of toxic genes, which is what gene silencing approaches are all about. And another approach is to edit defective genes. And these approaches have been investigated since the early 90s. And the transformative potential of gene therapy is really now evident. The first gene therapy was approved in 2017 in the US. And currently today we have 5 approved drugs, but there are now many candidates in various stages of clinical development. And you can see here 1,200 phase 1 gene therapy trials and phase 2 gene therapy trials. Some of these advanced to phase 3, and several of these are now reaching pre-registration, which is where you go just prior to approval. So in short, these gene therapies, which target the underlying cause or modifying causes of disease, encompass a one and done treatment, and they have tremendous power to relieve the burden of disease. Our approach to gene therapies for HD are based on adeno-associated virus, as Sarah mentioned. This is a cryo-electron micrograph of an AAV. And the key points that we like to consider is that the vector, or in this case, the AAV, carries that genetic material into the cell. So we deliver it into the body. For liver disorders, you deliver it into the blood. And we believe that for brain disorders, the best place to go for an AAV gene therapy is into the brain. AAVs that are very efficient at carrying their cargo into the cell means that less material is needed for therapy, which is safer for the patient. Now, what is the cargo that we're talking about in the case of the genetic material? This can include sequences that reduce proteins that impact HD. And in our case, for our Transformative Research Award, this is MSH3 and PIAS1, which you'll hear more about from Dr. Leslie Thompson. And because our work with AAVs and the AAVs that we've developed work in multiple species from mice to human cells, we can be better informed in translating our discoveries to patients. So what do I mean when I say our AAVs work in multiple species? And I've tried to highlight this here. So we work in mice models of Huntington's disease, but it's critical that our vectors work very well in those. And you can see from the blue color here, that means that the gene is being expressed. And this is sort of a reporter of the eventual cargo that we will use, which will silence MSH3 and PIAS1. And you can see in the mice, we can reduce expression in the deep brain areas and the cortical areas that are affected in mice models of HD. And importantly, because HD patients have cortical and deep brain manifestations, we can also target very beautifully and very efficiently those regions in a non-human primate, which is very close, of course, to a patient. And what we've discovered, given the potency of our tools, is that a single infusion per brain side can result in gene delivery to key brain areas impacted in HD. And you'll learn more about this from Dr. Jang-Ho Cha. And then finally, our vectors here shown, the reporter is now shown as more green than teal. And you can see very efficient gene transfer into human neurons in a dish that allows us to work with Dr. Thompson's lab in assessing the utility of our tools. So in summary, for my component, gene therapies for HD are promising because, one, we've discovered these really powerful new AAVs that achieve broad gene therapy to regions critical for HD patients. And importantly, this can be done at low doses and low volumes because they are so efficient. This then results in shorter surgery times, improves safety because we don't expose the entire body to the vector, such as the liver or the kidney or other tissues. And then finally, because a typical batch of drug made for clinical use can treat many more patients, it may have downstream improvements in terms of cost. We have the knowledge of key targets that when reduced via gene therapies may be beneficial to patients, and you'll hear more about that in the next component of our presentation. And we have the capabilities at the Center for Cellular and Molecular Therapeutics here at CHOP to make products for preclinical and early clinical studies, and the Transformative Research Award team that can test and advance these vectors from the bench to the work in patients. And down below here, I just show you one of the beautiful examples of the ability of our vectors to potently transduce cells in the cortex. And you can see those processes going down into the deep brain areas, which is an area that they communicate. And so with that, I'd like to pass the baton on to Dr. Thompson.
All righty. So yes, so I'm going to be tag teaming after Bev, and it's really been a wonderful journey to get to this point. Bev and Jang-Ho and I have been collaborating for many, many years, and it's really been wonderful to get to this point. Just showing all the work that's been done is really a consequence of the people in the lab who are shown here. We've had some new additions since this picture was taken. And as Bev talked about, sort of how I got here and where I came from. And so I was born in New York, and then very short stint to Atlanta, Georgia, where my sister was born, back to New York, and then to Wisconsin, Elm Grove, Wisconsin, little town, and played flute. And I probably would have gone through that path and stayed in music, except that we then moved to Mexico, to Guadalajara, Mexico for high school. And this is where really developed, I was able to learn Spanish, which comes into play a little bit later, and just decided that I wanted to go into something where I could impact patient care in some way, shape, or form. And I was very interested in research. I'd always been interested in research. Even as a child, have a microscope, chemistry set in the basement, things like that. And then my senior year of high school, moved to San Diego so I could get residency for college. Completed my undergrad at UC San Diego there, and then moved to UC Irvine, where I have been ever since then. You'll see a lot of changes with Bev and Jang-Ho, but I've been at UCI since I started graduate school. And in graduate school, I took a class. I was using yeast as a model system at that time. Took a class from John Wasmuth, shown here, who was a member of the HD consortium along with Nancy and all the labs who were involved. And I was just absolutely fascinated by this, that this was the time when Francis Collins had shown linkage to the cystic fibrosis gene. It was when the gene for Duchenne's muscular dystrophy had been discovered, and when the HD gene had been localized to a particular chromosome. And this was chromosome 4. And I show this high resolution physical map here, which John's group worked on through the Human Genome Project. That was a national endeavor to sequence the first human genome, which now we can do in a very short period of time. But at the time, this was a major endeavor to sequence a human gene. And like you see a physical geographical map here, this was a physical map of the human genome that he was working on. And I joined his lab as a postdoctoral fellow and have been working on Huntington's disease ever since. And I was fortunate to be part of this consortium. And this is a picture from Isla Morada, where we would meet once a year and share reagents, share the latest research. And my first postdoctoral fellowship was from the Hereditary Disease Foundation at that time. And this was just a fantastic, amazing thing to be part of. Really learned a lot from all these individuals. And this culminated, of course, with the identification of the disease gene in 1993. And because I was able to speak Spanish, I had grown up in Mexico, Nancy invited me to join her in Venezuela. And this is when I met Jang-Ho for the first time as well. We were both down there together and learned a lot about Huntington's disease, because there's such a large number of individuals and families who are impacted by this disease down there. Nancy's shown over here, and Ann Young, who was what she said about the neurology was the final word. She was always just amazing in her diagnosis of patients and understanding their symptoms. And we've had a large group of patients here in Orange County as well, and families that we've interacted with as well, which has really fortified this commitment I've had over the years to Huntington's disease in the hope of finding a treatment. And that's been really exciting now, working with Bev and Jang-Ho on this project. So wanted to get into the two targets that we're studying here. And as many of you know, I know that it's been shown that the age of onset for a given repeat length can really vary. And there were these pivotal studies from the GEM Consortium and other labs where it was found that the modifying genes that might be able to slow or accelerate HD onset may be therapeutic targets. And many of these fell in the category of DNA damage repair, where the DNA itself is prone to expansion, the CAG repeat. So I'm going to show you that in a minute. But what this means is that you have individual variation. So this would be somebody that has sort of classic age of onset between 40 and 50, say, and then this variability at a given repeat length. And so let's say someone has 45 repeats. A modifier that's protective would extend that age of onset to be later in life, whereas something that would be a negative modifier would extend it to earlier in life. And so we want to really target those modifiers that are protective and reduce those that are deleterious, that might have earlier age of onset. So what does this mean? And what is responsible for this? So we've learned over the years that what may be really pivotal in all this is what's called somatic repeat expansion or somatic expansion. And this means that an individual starts with a certain number of repeats when they're born. It's in all of their cells. We have Huntington in all of our cells. And during aging, what can happen is that in the brain, in an individual who carries a repeat expansion that causes Huntington's disease, can expand. And they can expand to very long repeats. And this slide is courtesy of Vanessa Wheeler, who's been instrumental in finding this and documenting this repeat expansion in the brain. And so it starts to grow in an individual. And this is just primarily in the striatum, in the region of the brain that's most vulnerable to Huntington's disease. And it can expand to very long repeat lengths in that tissue. So in the skin or in the blood, it's still, say, a 40 repeats. But in the brain, it starts to expand. And that expansion seems to be very important for when symptoms start to progress, onset of symptoms, et cetera. So we're really interested in targeting this. And there's been a number of publications out, including from Vanessa, Sarah Tabrizi, the GEM Consortium, and Christopher Pearson, where they found that a particular DNA repair protein, MSH3, is required for these CAG expansions in the brain in both mouse models, and it's a human modifier. So in this case, MSH3, you want to target it and reduce its activity, or it will lead to earlier onset. And to show this more graphically, this is just from a mouse model of HD. And what you can see is in the tail, for instance, of a mouse, you have the standard repeat length. And this can have some variability, not a lot, but some variability around, say, this 103 CAGs that's inherited. But in the striatum, in this part of the brain that's affected, this grows. And this is this repeat expansion I was talking about. These are different bars, represent different CAG repeats. So it gets longer and longer and longer. But in a mouse where it's reduced partially, MSH3 is reduced partially, you don't see that long tail, as much of the repeat expansion. And in a mouse where they don't have any of that MSH3 expressed, you don't see that at all. So you prevent that repeat expansion. So that's why we're targeting that particular protein. And the second one is PIAS1. And this is in this whole area of SUMO modification, which is small ubiquitin-like modifier. And basically, it's a SUMO group which is shown here, the crystal structure shown here, it's just a very small protein, can be added to a given protein in a cell and then taken off. And this is extremely rapid process. So when these are added, they're very quickly removed. So it's very highly transient shown here, but it's very critical in a cell. It maintains the balance of protein clearance, it maintains transcription, expression of RNAs, who a protein talks to, so the interactions that occur. It's involved in this DNA repair process that I was just mentioning, and it's very important for the synapse, for communication between neurons in the brain. And in 2004, Joan Steffen, who's an associate professor in our group, had discovered that Huntington, the protein itself, can be sumo-modified on multiple sites in the protein. And then from there, Jackie O'Rourke, who was a graduate student at the time and now works for Ionis, discovered that a protein called PIAS1, protein inhibitor of activated STAT, could enrich that sumo modification. So it could enhance the modification by sumo of the Huntington protein. And we also found that proteins that have this sumo tag start to accumulate in the HD brain, particularly in the striatum. And if you can reduce the expression of PIAS1, you resolve that. You don't have this aberrant accumulation that reflects that the garbage is not being cleared out of the brain correctly. And so this is where we started working with Bev's lab, and a graduate student, Joseph Ochaba, had done a study where we used Bev's virus, injected it into the brain to reduce PIAS1. And if you look at the wild type mouse, so just treated with PIAS1 knockdown or untreated, and this is a pole test where they just climb down the pole, and an unaffected mouse will just scoot down very quickly. The HD model, so this is where the HD mutation has been put into the mouse, they have a much more difficult time climbing down this pole. They kind of scoot down slowly. But if they're treated with this virus that reduces PIAS1, they look very similar to the wild type mice. And it really prevents many, many different mouse symptoms, including this pole test. So what we know now from a number of different studies over the years, and I haven't cited them all, but half the lab has worked on many of these, is that when we knock down PIAS1 in a number of different mouse models, again, using the virus from Bev, we see improved motor skills. We've reduced this aberrant Huntington accumulation. It rescues the transcriptional changes that are observed, and it improves DNA damage repair in a pathway that's very protective to neurons. And we've done this also in induced pluripotent stem cells. So these are cells that are derived from patients, reprogram back to a state that you can differentiate or guide them into any cell you want. But we've studied this in neurons, and we see this same type of restoration of, say, synaptic transcription, improved DNA damage repair, and even the way proteins go in and out of the nucleus was repaired. And finally, there's a variant that's associated with later onset of HD. So there's a change in the DNA in PIAS1 in some individuals where they have a later than normal onset, which means this is protective. And that particular change in the DNA causes PIAS1 not to work as well. So somewhat the equivalent of what we're seeing when we knock it down. So even in the human situation and in sort of a genetic case, we also see that there's protection from reducing the activity. So this has huge potential for therapeutics for both MSH3 and PIAS1. And our question here for the TRA is that what if you can reduce protection of MSH3 and PIAS1? So if we lower those each separately or together, see if you get an added benefit if you can reduce both of those at the same time, and together with viral vectors that are developed at CHOP and in Bev's group. And would this rescue HD symptoms, prevent this somatic repeat instability and other symptoms? So we will be testing this in the iPS neurons and mouse models of Huntington's disease. So next, I will hand it off to Jang-Ho, who will talk more about how we're going to do this.
A real pleasure to be with everyone today, and thanks for the fantastic introduction from Sarah. And also you see the star power from Bev Davidson and Leslie Thompson. We've known each other for years, and the ability to collaborate on a program such as this is so exciting. And so we're happy to bring this to the larger audience. A little bit about my own journey. I was a Harvard undergraduate and really fell in love with the brain. I went to school at Michigan where I got my medical degree and my PhD degree. And Ann Young, who you've seen, was my mentor and really raised me. But since that time, my playbook has been pretty simple. I'm a science nerd, and the idea is to use science to help develop a treatment for people with neurologic diseases. And it was because I was in Ann's lab that I was first able to participate in the Venezuela project, where I did meet Leslie Thompson. And my job initially was to lift heavy things, but it really got me very hooked in the HD population to see a disease that was so devastating. And also the idea that I could spend my scientific career to make a dent in this disease. And then I went on to pursue neurology training, and then on the faculty at Mass General Hospital, Harvard Medical School. And I really focused on neurodegenerative diseases such as Huntington's and Parkinson's and Alzheimer's. Along the way, the HDF has been really important to me. In fact, my first fellowship I ever got was from the HDF, and I also had the pleasure of serving on the scientific advisory board for many years alongside Bev and Leslie. And I only got off of that board when the company I was with at Novartis had a HD program. But I've also been involved with the Huntington's Disease Society of America, which is, as people know, is the patient organization. I did move to Merck to learn how to develop drugs. And then for 8 years, I was at Novartis, based in Cambridge, Massachusetts, where my group oversaw all the early development for drugs for neurology conditions. And then I joined Latus Bio. This is a misprint in December 2022, not 2020. So what is Latus Bio? So this is a company that was founded by Beverly Davidson and the research that came out of her lab. And again, all the people in the bottom picture are the ones who have done the work. But it's really taken this idea from the bench that we could have much better virus delivery systems for gene therapy with the focus on CNS indications or CNS diseases. And so we really think we've got the best viruses. I'll show you some data which lead us to believe that. And this all of a sudden makes it possible to consider that we could develop really transformative treatments for some of these worst diseases, including Huntington's disease. The people in Latus are very committed to Huntington's disease. So Bev is our scientific founder, and you've heard all about her, and she's been on the scientific advisory board of the HDF. I've really devoted my career to Huntington's disease, and I've also had the pleasure of serving on the SAB of the HDF, as has Sarah Tabrizi, who is our advisor and on our scientific advisory board, but she is a well-known world-leading clinician and also has served on the SAB of the HDF. George Yorling, who's our executive director of development, spent many years at HDSA. Previous to that, he was at CHDI, and he himself was, when he was a postdoc in my lab, was the recipient of a John Wasmuth Fellowship from HDF. And we also have Paul Ranum and David Lee, both of whom were, when they were in Bev Davidson's lab, were recipients of HDF Young Investigator Award. So I think we are really indebted to the HDF for developing these scientific careers. Let's talk about the task at hand. In Huntington's disease, we know that there are specific areas that really are particularly affected. And in the brain, there are the deep parts of the brain. You've heard about the striatum or the caudate and putamen. So we know that if we're going to cure Huntington's disease or really have a treatment that makes a big difference, we have to be able to deliver the gene therapy to these areas. And this has been a challenge in the field, getting viruses that can deliver gene therapies to these deep areas of the brain, which are so important. The current viruses are not going to do it. So here's an example of the conventional virus that is most commonly used for gene therapy. It's called AAV9. And this is a picture of a monkey brain, and where the AAV9 has gotten into the brain is represented by a brown color. And you really have to search to find these small areas. And the remainder of the brain section that you're looking at under the microscope, you're not getting sufficient entry of the virus. And so if you look at a virus like this, you know that it's very unlikely that you're going to be able to deliver sufficient amount of gene therapy to correct the disease. That's not going to be sufficient. And so when Bev first talked to me in 2022, she started showing me pictures like this. You've seen some of these pictures before, but what you see is in the striatum, again, this is the area of the brain that is most affected in Huntington's disease. You see on the left all these little dots. Those are indicative of where the virus has gotten into. And so if you look at this, and on the right, you see it higher magnification, and these are the outlines of brain cells called neurons. So you have a high degree of confidence, actually, that you can get into the right cells in the right structures. This is the striatum. And as I tell people, the 3 rules of neurology are location, location, location. So what we know is with the conventionally available viruses, they don't get into enough cells into the right places that you need to cure this disease. But with the Latus capsids, now we see, wow, we're really getting a lot of these brain cells in this most important region of the brain for Huntington's. An interesting thing is when you inject it in one area, these viruses tend to spread to many different areas of the brain. And so we have an injection in one area, but you can see if you look far away, this is the cortex, which is the outer part of the brain. This is the caudate, which is the deep part of the brain. This is the thalamus. You don't need to know these terms, but what you do need to know is these are different areas of the brain, and we haven't done 6 different injections. This is from a single injection. So it looks like these viruses, once you put them into the brain of a monkey, which does get as close as you can to the human brain, tends to disperse. And so when you think about what's the type of gene therapy we want to deliver, this starts to become some of the profile of what you need to get it there. Location, location, location. Even further out, so if you look at, if we inject deep into the brain, what we can see is the cortex, and this is the outer part of the brain, is pretty labeled pretty well. That means these viruses are traveling to areas that are really far away from where you directly inject them. And we think that this is really important as we think about Huntington's disease therapeutics. We know that the deep areas are the most affected areas, but we know that it's not the only part of the brain that's affected. And we know that the cortex is also affected in Huntington's disease. So we think that this distribution of the viruses is really permissive for thinking about a gene therapy. So because these viruses spread so well, we think that we might be able to get by with just a single injection on each side of the brain, as Bev has alluded to. I think people are aware that there are current HD gene therapy approaches, which in order to achieve the spread throughout the brain, multiple injections occur. And this can result in really a prolonged, very burdensome procedure, up to 16 hours in the operating room. But we think that with these kind of new and improved capsids, we can do this with a single injection on each side and cover large parts of the brain. So how do we get this into patients? There are a lot of steps that any drug has to do. First, we have to test in cell and animal models that we can knock down these targets. And you've heard Leslie talk about MSH3 and PIAS1. When we get the right gene therapy to do that, we call that our development candidate. And then we'll take that development candidate into a mouse model of HD, say, okay, now does it work in a mouse? Then we'll see, okay, is it safe? And we'll test the safety in a large animal model. And this safety data are really important because that's what we'll bring to the FDA or other health authority in Europe, let's say, to demonstrate that we have confidence that we can bring this into people with an understanding that it will be safe. And when one starts clinical trials, the first trials are really about safety. And then the second clinical trial is, does it actually work? And so each of these steps kind of take a lot of time, but these are the steps that we are marching through as we speak and we're planning for now. And so we have a lot of work ahead of us, which is a good problem to have. Just ending with where it started for me, here's my mentor, Ann Young, and Nancy Wexler examining patients in the barrios of Maracaibo, Venezuela. Here's skinny me in the middle. And then here's a relatively recent picture of me and my good friend, Nancy Wexler. And with that, on behalf of our whole team, I just want to thank not only the Davidson lab, not only the UC Irvine lab, but everyone at Latus Bio, and especially the Hereditary Disease Foundation for making this visionary grant make these things possible.
Thank you all so much. Thank you for presenting that super exciting work. Because it was so exciting, we have a lot of questions, so I hope that we'll be able to get to them all. But everyone, keep submitting your questions in the Q&A box. Please use the Q&A box, not the chat box, throughout the rest of the Q&A session. We'll try to get to as many as we can. Again, they can be technical, non-technical, just any question you may have for our speakers. So the first question I'm going to ask was actually asked by someone in an HD Facebook group that I'm part of. He can't join the webinar today, but he's going to watch the recording and he wanted to make sure this was asked. So this is a question from Wang Chi Yang. He wants to know, what is the relevance of the advancement of HD gene therapies for some of the other CAG diseases, such as SCA1 and 3? So I don't know who wants to take that.
I think Bev.
Yep.
Yeah, so the manner, thank you for that question. The manner in which we, there's two components of this, right? There's the capsid component, and then there's the cargo component that we talked about. So I think for the cargo component, what we're testing here could definitely have relevance for the other CAG repeat expansion disorders. And we do need to understand better how much that expansion is critical to the pathogenesis in the regions and cells impacted in the spinocerebellar ataxias, for example. And then with regards to the vectors, similar to the highly potent tools that we've developed that Jang-Ho and I showed, we have similar programs for improved potency for the spinocerebellar ataxias, where regions of the cerebellum are really critical to get that cargo into.
Thanks, Bev. So we have a question from an anonymous attendee about delivery methods. So maybe this one could go to Jang-Ho. They ask, can these AAVs cross the blood-brain barrier to achieve less invasive methods of gene delivery rather than brain surgery?
Yeah, it's a great question. So lots of companies are exactly looking at delivering intravenous AAVs, and specifically for that reason. But what we're seeing is in gene therapy, you have one of the limiting factors is toxicity. And a lot of that toxicity is not from toxicity in the brain, but in other parts of the body, such as the liver. So what we know is the majority of viruses, if you put into the bloodstream, actually don't go into the brain. So in order to combat that, what companies have tried to do is inject huge amounts into the bloodstream, but most of that blood flow actually doesn't go to the brain and goes to the liver. So we have opted for a more direct approach. This is a brain disease. We figure we can go directly into the brain. And especially for a one and done therapy, we think that in order for it to work, it has to be in the brain. And then if you deliver the viruses to the other parts of the body, you don't get any better efficacy. It doesn't work better, but you can get a lot more problems. And there have been lots of problems with peripheral organs, including death from immune responses. So we think the better part of valor is to put it directly in the brain, especially for a gene therapy that could be a one and done.
Thanks, Jang-Ho. So I think I'll send this one to Leslie. This is from Caroline Ben. She wants to know, is MSH3 simulated? And if yes, does PIAS1 influence this?
So we've done some assays to test all of the or a number of the DNA repair proteins. MSH3 does not seem to be sumo modified, but many of the other ones are that are involved in various complexes. And we're checking whether they're sumo modified by PIAS1. Thanks, Caroline.
Thanks, Leslie. So this is again from an anonymous attendee. I'm not sure who wants to take this, but they ask, what phase of the disease is this gene therapy targeted for?
Yeah, I'll take this. I think the first phase of experimentation is always going to be at safety and tolerability. And so we'll need to understand kind of for what population the risk benefit is the best. When you do get into a clinical trial, there's a couple of competing considerations. For any therapy, you want to start earlier because there's kind of more brain to preserve and you might have a bigger effect. But the way we follow the clinical status of patients in clinical trials depends kind of on symptoms. So it's kind of harder to tell if your treatment is working if you're starting off with patients who don't have symptoms. So that's kind of the push and pull of should you go to an earlier population or a later population.
Thanks, Jang-Ho. Maybe I'll send this one to Bev. An anonymous attendee asked, will any of the gene therapies reduce and reverse symptoms? Maybe you could talk a bit about what you've seen in preclinical models.
I think that's a great question, and thank you for asking that. There is no reason to think that the therapies wouldn't reduce the further progression of symptoms. And the question is, as you go earlier and earlier, you may be able to treat before there are any symptoms whatsoever. Whether or not we can go into later stage patients and see reversal of symptoms, I think is an experiment that just has to be done. But I think that the animal models proved to be very promising, and particularly the cell model systems that Leslie is working on, I think are really giving hints that there is some reversibility to these symptoms, at least in the cells. So the cells are showing us signs of disease and they're still alive. And when we correct that, then there is reversibility. So we're hopeful.
Thanks, Bev. So I think this one could go to any of you, but I think I'll send it to Leslie. This is from Yinqing Wang. They want to know what's unique about the striatum that promotes somatic instability in this specific tissue type.
Yeah, I wish I knew the answer to that one. I don't think it's really clear. Seems to be very vulnerable, and people like Nat Hines and Stephen Carroll are really investigating this quite deeply to understand what the pathways are that might be responsible. But it does seem to be occurring in especially medium spiny neurons in the striatum the most. And that includes, I won't get into that, but even in some of the other disorders, you see somatic repeat instability that's largely confined to the striatum, such as SCA1, work that Harry Orr has done.
Thanks, Leslie. So I'm going to send this one to Jang-Ho because in the chat privately, he offered to take unique questions. So this is from an anonymous attendee who would like to know, why is the company called Latus?
Oh, I'm going to toss that right back to Bev because it was named Latus before I got there.
Latus is Latin for broad, and the idea behind Latus is that we've developed a platform for discovering vectors that can find a broad range of applications. And some of our vectors have very unique properties that allow use for many different disorders.
That's great. I like that a lot. Thanks, Bev. So I might add, so this one is to Jang-Ho, and this is a science question. So this is from Simone Moore. They ask, what percentage of neurons are transduced in key regions?
Yeah, it's a great question, and I can't disclose all of that, but I can tell you what we're doing is we're injecting and we're quantifying, and depending on the vectors, what we see is in some regions 70 to 90%. In other regions, we're seeing 50%. It depends on the vector that we're using and the quantification methods. There are lots of questions that we have in terms of, can we get a higher percentage of neurons transduced? But I can say with confidence, we're seeing a much higher percentage than existing parental serotypes. So exactly tracing down that number is kind of an area of active investigation. Thanks,
Jang-Ho. And I just saw one that I wanted to ask, but it went away in the queue because I think Bev said she wanted to answer it live. But this is from Mark Deecher. They ask, how long do you think the therapy will work in the body? And could the brain create a, quote, workaround over a long period of time? That's an interesting concept.
Yeah, thank you, Mark. I started to type the question and then realized I said answer live. So sorry about that, Sarah. So in human patients that have received these kinds of vectors and derivatives of these kinds of vectors, especially in the brain, there hasn't been a diminution of gene expression over many, many years. And so as long as the cells are alive, we should see robust long-term expression. You do see loss of durability in peripheral tissues like the liver, and this is largely, again, due to the immune responses from these very, very high dose therapies that are delivered there. So we think that the body may not create a workaround here, given what we've seen in animal models and in cell model systems. And again, the robust durability of these vector systems in the brain really provides for, as Jang-Ho said, a one and done therapy.
Thanks, Bev. So I'll send this next one to Leslie. So this is again from Carol Ben. She asks about the homology between MSH3 and PIAS1 in mice and humans, and asks if you're using a sequence that will target in both species or if you're going to have to make humanized mice.
Yes, so they are actually quite similar. And as Bev can say, we're using the same virus for both. So it's the same target.
Thanks, Leslie. And there's kind of a few questions with the same flavor. So they're not from anyone in particular, but people are curious, when is this going to get into clinical trials and kind of the timeline that you guys see for this type of approach with your new AAVs going to the clinic?
Let Jang-Ho handle that one.
Yeah, I mean, we want this to happen as soon as possible, but there are steps that we have to do, and all these steps take time. So I would say it's on the order of years. It's not months. It's on the order of years, but we have, laid out kind of the sequence of events, which we had to do when we submitted the grant. But we are very hell-bent on getting into the clinic. That is, in getting into people. That is the only goal of this research. We're not trying to create interesting science along the way. So even with that focus, things do take time, and properly so. We have to really ensure that we understand everything about safety in a prolonged animal safety experiment, and really understand every single possible safety issue that we might encounter in the clinic. So I'm going to say it's more than a year, but I can't get more precise than that.
Thanks, Jang-Ho. I think that's an important point, right? We want to make sure we can get these to people, but also that they're safe. And so I think that might be a nice concept to wrap up on, because it looks like we're at the end of our time here. So if you had a question that we didn't get to, which there were a ton of questions that we didn't get to, I will send those to our speakers, and hopefully, if they're willing, they'll follow up with you directly. So I want to end by again thanking our sponsors for this webinar, Neurocrine Biosciences and Spark Therapeutics, as well as everyone who donates to the HDF to support the research that we fund. Of course, thank our audience, and last but certainly not least, our speakers. Thank you all so much for joining us today. Our
Can
next
I say
webinar is
thank you
thanks,
so
Guy.
much to Nancy and the HDF, and
Oh,
thank you, Sarah.
yeah, absolutely. So our next webinar is Tuesday, May 14th, and will feature an update on clinical trials. We are going to be joined by Dr. Ed Wild from the University College London and Dr. Frank Bennett from Ionis Pharmaceuticals. They are going to be talking about getting drugs from the lab bench to the clinic, and will summarize what's happening now in HD Therapeutics. Thank you all so much for joining us, and we hope to see you at our next webinar.